I am going to chair the first session, which is on radiation resistance, and our first speaker is Graeme Hewitt. Graeme is about to start his group. He's a junior group leader within RadNet City of London, and the title of his talk is Targeting the DNA Damage Response in Combination with Radiotherapy. Thank you, Graeme.
So, hello, I'm very excited to introduce myself to you all today, especially since it's the first talk that I've given in person for well over a year. So, my name is Graeme. I have been working in Simon's lab as a postdoc on DNA damage repair, and today I'm going to tell you a little bit about my background in working in DNA damage repair and how I'm going to apply that to radiation biology. So my background is I started to work on DNA damage as a driver of cellular senescence and aging during my MRes and PhD at Newcastle University. I then moved to London to the CRUK into Simon's lab, where my research was much more focused on basic mechanisms of DNA damage repair. And very nicely, coinciding with my time in Simon's lab, Artios Pharma was founded and has rapidly grown. And Artios is a CRUK spin-out company based on targeting the DNA damage response to treat cancer. And I've been involved in a number of different projects with Artios. During my time in Simon's lab, I also set up platforms to do CRISPR screens to dissect DNA damage repair dependencies. And it's these two things, these two last points, which I'm going to talk to you about today, and how I'm going to apply them in my new lab to interrogate different things in radiation therapy. So targeting the DNA damage response in cancer, mutations in DDR genes are very common in cancers, and they drive genome instability and tumorigenesis. The good thing about this is loss of one DNA damage repair pathway can also put a critical dependency on another, and this is termed synthetic lethality. So this is just a schematic to illustrate synthetic lethality here. You can see at the top here, in a normal cell, you have both genes expressed, they're fine, they're viable, and loss of one gene or the other doesn't necessarily lead to lethality, as it can be compensated for by the other. But if you were to lose both genes or lose one gene and inhibit the other, then it leads to lethality. The same can also be true for genes which are overexpressed, where it puts a critical dependency on another pathway. So this is best exemplified by the success of PARP inhibitors in the treatment of homologous recombination deficient cancers. So with the instance of PARP inhibitors, in a wild-type cell, a single-strand break or a base lesion can be repaired in a PARP-dependent manner through base excision repair or single-strand break repair. However, upon treatment with a PARP inhibitor, these pathways are lost, and it puts a critical dependency on HR for survival. So BRCA1 and BRCA2 are common mutations in cancers, and they lead to HR deficiency. This is a slide taken from the SOLO-1 clinical trial of the PARP inhibitor olaparib in ovarian cancers. So these are patients which have either BRCA1 or BRCA2 mutations, and they've been treated with this PARP inhibitor. And you can see that the outcomes for the PARP inhibitor is a lot better than the placebo. So despite the success of PARP inhibitors in the clinic, there's a big problem with resistance mechanisms. And one of the tools which we can use to interrogate these resistance mechanisms are CRISPR screens. And this is one of the things which I set up in my time in Simon's lab. So the schematic on the left here is a very simple version of what we do in these screens. You take a cell line which expresses Cas9 or has inducible Cas9, you introduce a library of guide RNAs for the genes which you're interested in this case, a whole genome library, and then you expose your cells to your different treatments. And the readout at the end are looking at the counts of guide RNAs by next generation sequencing. So loss of a particular guide RNA represents a gene which, the loss of this gene leads to lethality. So on the right-hand side is just a scatter plot taken from my last paper, where we looked at olaparib treatment in wild-type cells. And encouragingly, you could see known resistance mechanisms such as loss of PARG and PARK1 come up on the right-hand side. But interestingly, we found this chromatin remodeling factor, AOC1, leads to PARP inhibitor sensitivity. So to cut a very long story short, using these screens and different techniques, we're able to identify that loss of AOC1 not only leads to PARP inhibitor sensitivity, but it also leads to synthetic lethality with HR. On top of identifying this synthetic lethality, these screens also help us identify the endogenous lesions which were responsible for this phenomenon. So this put AOC1 at the forefront of being a novel DDR target, and this is with the publication of my most recent paper, and also 3 very similar papers at the same time, which came to very similar observations. So at the same time as trying to find novel DDR factors that we can target in cancer, I've also been working alongside Artios with a number of their different programs. And previous genetic studies have shown that polymerase theta is an attractive target in the treatment of HRD cancers. So at the moment, Artios have developed potent small molecule inhibitors against POLQ. And POLQ is a particularly attractive target as it's not highly expressed in somatic tissue. So this helps greatly mitigate some of the problems that you might have with toxicity. So this is just a little snapshot of the most recent publication where these inhibitors were first published. And you can see that treatment with POLQ inhibitors leads to the death of BRCA2 deficient cells. So these are HR deficient cells. So the inhibitor can phenocopy what we see genetically. And interestingly, or at least more interestingly to us, inhibition of POLQ also leads to radiosensitization. So treatment of wild-type mesh with POLQ inhibitor leads to sensitization similar to what you would see with a genetic knockout, whereas treatment with the inactive control, you do not see this. So if anyone's interested in learning more about POLQ, this paper was just recently published in Nature Communications. But what I would like to highlight is that this was a big team effort, and it shows that when academia and industry work together, we can do some pretty cool things. So the last 2 points are the most important. The POLQ results in radiosensitization, and as I said before, that it's not expressed in somatic tissues. So this helps mitigate toxicity, which is a common problem with targeting DDR in combination with radiotherapy. So what is it that I'm going to do in my new lab? So one of the projects which I'm in the process of working on is to perform whole genome screens to interrogate the resistance mechanisms and sensitivity mechanisms in this combination therapy of POLQ inhibitor with fractionated radiation. On top of this, I also plan to look at different modalities of radiation, such as proton beam, and see if the resistance and sensitivity profiles are similar between different modalities. So very quick whistle-stop tour of what I'm planning. This is just the acknowledgement. So there's a large number of labs that were involved in the work that I've just shown. And I'd like to give particular thanks, of course, to Simon for supporting me throughout my postdoc, as well as Artios for the collaborations that I've had during my postdoc and will continue to have in my own group. And of course, many thanks to Cancer Research UK, RadNet, for giving me the opportunity to start my own group.
Do we have any questions? Anything online?
No online.
Anyone in the room? Don't be shy. Oh, Kebs. Oh, okay, super. Can you come up to the mic? That would be great. Thank you.
This is really more out of curiosity, but where is POLQ expressed?
So it's expressed predominantly in the thymus. So the data on expression levels have been done mainly with looking at RNA-seq. We're currently trying to develop better tools in order to look at the expression, so mainly nanobodies and different antibody approaches in different tissues. So yeah, we're working on it. But looking at mRNA expressions, the thymus seems to be where it's mainly expressed.
And is it essential there in the thymus?
No.
So I've got another sort of expression question as well. PARP inhibitors,
Yeah.
Do they affect the stroma, the tumour stroma?
I would imagine yes. I don't know. It's not something I've looked at personally, but yeah, they would affect the stroma for sure.